Hello everyone and welcome to today's webinar, 7 Steps for Statistical Success in Clinical Trials, Good Clinical Practice Guidelines, Part 2. Before we begin, I would like to show my respect and acknowledge the traditional custodians of the land, of elders past and present, on which this meeting takes place. I would like to go over a few items so you know how to participate in today's event. You have joined the presentation listening using your computer speaker system by default. If you prefer to join over the telephone, just select telephone in the audio pane and the dial-in information will be displayed. You will have the opportunity to submit text questions to today's presenter by typing your questions into the questions pane of the control panel. You may send in your questions at any time during the presentation. We will collect these and address them during the Q&A session at the end of today's presentation. If your questions are unanswered due to time limitations, we will upload answers to these on the ACTA website. Today's webinar is being recorded and you will receive a follow-up email within 24 to 48 hours with a link to view the recording. I would now like to introduce our presenter. Welcome, James. James has over 30 years of experience as a statistician in the pharmaceutical industry. He has led the development of early clinical trial design, decisions and analysis, and the development of regulatory reimbursement approvals for biotechnology products involving company representation at regulatory submissions in the US and EU. James has been a member of the Society for Pharmaceutical Statistics, the PSI, for over 30 years, serving on the scientific and training committees, as well as the board of directors. He has also served as the Professional Affairs Committee of the Royal Statistical Society, where he has been a chartered statistician for over 25 years and is currently a member of the Scientific Committee of the European Federation of Pharmaceutical Statistics. In 2020, James moved to the Strategic Consulting Group in Cytel. I wish to welcome you warmly, James, and I will now hand over to you. Thank you.
Okay, thank you very much, Anisha, and thank you very much to the Australian Clinical Trials Alliance for inviting me back to give part 2 of this presentation on 7 steps for statistical success in clinical trials. As Anisha said, my name is James Matcham, and here is my disclaimer, if I can get the slides to move. Here we go. I am an employee of Cytel, and the views expressed in this presentation are the views of me, not necessarily the official position of Cytel. And if you remember, this is part 2 of a 2-part training, and the objective of this course is to introduce, refresh, or update your knowledge of ICH and other international guidelines as we go about designing, conducting, and analysis to clinical trials that we work on in our day-to-day jobs to ensure they're providing high-quality work to international standards. So if you attended the last meeting, the agenda for the whole training course looked like this, and in the last meeting we did quite a bit of this agenda. So we started off with an introduction to GCP, and we talked about the ICH framework and about how each ICH guideline goes through different steps of development. There were 5 steps to do that. It is done by an ICH international group, and we talked about that group as well. And then we gave quite a high-level review of 5 of the ICH documents. So we started off with Good Clinical Practice, which is ICH E6. We then moved on to the Biostatistics Guideline, ICH E9, and its revision, and the Estimand revision, which we're all busy trying to get used to right now. We then moved on to Advice for Selection of Control Groups, that's ICH E10. And then we talked about multi-regional trials, and that's ICH E17. So that's where we got to in the last training session. And what we're going to do in this session today is talk about additional guidance. And the number of additional guidances that are around from the big regulatory boards. I'm mostly going to talk about additional guidance that's provided by the FDA in the United States and the European Medicines Agency in Europe. I'm also going to talk about the number of additional guidances that have come about because of adaptive trials. And we'll talk about adaptive trial guidance towards the end of the session today. At the end of today, there should be about 10 minutes for questions, so please hold your questions till then. And if we don't get time to answer all of your questions, please submit them anyway, and we will provide written answers on the ACTA website. So, additional guidance then. There are two main sources of additional guidance. The first one is European guidance, and the European Medicines Agency provides quite a lot of guidance documents on their website. I'll give the website on the next slide. And they come in different forms. So they come in terms of points to consider documents. So points to consider in applications with meta-analysis, for instance, and one pivotal study. They also do reflection papers. And reflection papers come about if there's something new that they want to discuss in a regulatory sense, so that when you are filing clinical trial plans or clinical trial submissions to them, that you at least know ahead of time what you need to be discussing. So, for instance, there's a reflection paper on statistical methodology for comparative assessment of quality attributes in drug development. So this is the quality of materials. And then there's also one on methodological issues in confirmatory trials with an adaptive design. So, for instance, this is a relatively new topic, and so gets a reflection paper. Occasionally, they issue a qualification opinion. And there was a group in the pharmaceutical industry who put together a submission based around MCP mod, which is a methodology for dose response analysis. And they put forward a statistical methodology for model-based designs and the analysis of phase 2 dose-finding studies to the EMA. And they then awarded them a qualification opinion, which means then that methodology is admissible in those trials. So these are three types of European guidance. There's a few more, and here's one, guidelines. So typically, the process for European guidelines is there is a paper that's produced saying that there is a need for a guideline in a particular area, and that invites comments. They then produce a draft guidance, and the draft guidance then is open for comments from industry and other groups. And then some little while later, that becomes a final guideline. And this process is in motion, and we have some guidelines, some draft guidelines. A lot of guidelines stay in draft for quite a while, but actually the EMA are getting quite good at finalizing their guidelines right now. Some of the more recent guidelines documents are actually a really good read and contain a lot of information with examples and training and so on. So it is actually quite a good place to go and look for just general advice in statistical methodology in clinical trials. And you can see some of the guidelines here that are relevant. In addition to this, EMA provide a lot of disease-specific guidelines. So this would be for submissions in particular diseases where they have specific guidance about which endpoints to use, how to define your endpoints, which randomization stratification factors to use, and any particular guidance around diseases. And there's a large number of those. I don't have time to go into all of those today, but if you're working in a disease where there's a drug on the market, there's likely to be a guidance for that disease that exists on the European Medicines Agency website. So please, use that link, and that takes you to a search area on the EMA website, and you can search for methodological guidance and disease-specific guidance. So that's one area to look at, which is the European guidance. There is another area, which is the US FDA guidance area. And the US FDA take quite a bit of time to go from draft guidance to final guidance. Nevertheless, as soon as a document gets into draft guidance, most pharmaceutical companies look at that and treat it as final because that's what the reviewers are thinking. There are lots of different categories of guidance and hundreds of different guidance documents. We, of course, will focus on the statistical ones, but just like the EMA, there's guidance around much of the different things that you want to do in pharmaceutical trials, including disease-specific guidance as well. So you'll often find if you're planning a trial in a new area, a good thing to do is to get onto the European and the US guidance searches, search out the guidance and give them a really good read, because that tells you what kind of guidance people in the pharmaceutical industry are getting when they're designing their trials in those areas. So I think those two websites, the EMA one and the FDA one, they are probably really good for you to click on and go and play and search for guidance. So if you are working in a specific area, you're using particular techniques, search for those on those websites, and it will give you at least the guidance that these bodies are giving pharmaceutical statisticians for conducting those trials. So in the first part of this session, I'm just going to go over additional guidance. And what I've done is I've looked through the EMA and the FDA guidance and grouped the guidances according to topics. So the topics I've chosen are development planning, meta-analysis, non-inferiority, baseline covariance, data monitoring committees, and multiplicity. There's a typo on here. I'm not going to talk about endpoints, but that didn't make the cut. Just to say that just this last couple of weeks, there's been a new guidance issued by the EMA on the use of real-world evidence in the planning of clinical trials. So I didn't really have time to put that one in, even though I think it's probably a good one for you to look at. So if you're interested in the use of real-world evidence in the design of clinical trials, then actually that's just come out in draft and they're inviting comments right now. So this is a pretty fast-moving field. Okay, so let's turn to the first of these, which is development planning. When you're planning the development of a new product, there are some general guidances that come out, and between the two main groups that give guidance, there is advice on the amount of efficacy data you need, the amount of safety data you need, and minimums for the amount of patient exposure that you need to have in your filing. If you are in an orphan drug, a rare disease area, usually the normal rules do not apply, but there is some guidance around rare diseases in development planning. For instance, the FDA have a guidance document around development in rare diseases and gives some quite specific guidance in there. There's some guidance around combination products. So if you're in the world of oncology right now, almost everything is a combination product. And you can see some of the guidance for how to develop combination products. And there are lots of others as well. So there are two parts that I'll focus in on here. One is the amount of efficacy data, and one is around combination products. So in terms of efficacy data, there is one FDA document, which is quite an early document, about providing clinical evidence for the effectiveness in human drugs and biological products. And in here, you'll see some words that are used. Independent verification is needed. That usually means that you need 2 phase 3 trials, or 1 phase 3 trial and another large trial, which could be a phase 2b study, but independent verification is needed. It says that more than 1 adequately well-controlled trial is needed. Now, in the Speaker guidelines, this comes up quite commonly. Adequately well-controlled has a specific definition, and usually means randomized, usually means blinded as much as you can, and predefined and so on. So adequately well-controlled is a term that you'll see quite a lot. We will also need to minimize unexpected bias and variability. They're very concerned about the possibility of a false positive trial. And most interactions with regulatory bodies, you often discuss type 1 error quite a lot, and about minimizing the type 1 error and keeping that controlled to specific levels. So the possibility of a false positive trial means that there's a lot of guidance around multiplicity and control of type 1 error. And many of you will be aware of the many statistical methods that are out there that are concerned around false positives and keeping them at acceptable levels. Development planning also talks about your trial, if it's an adequately well-controlled, is likely to be in more than a single site, and talks about the rare situation of when there's scientific fraud and about what to do there. It also highlights when you need to discuss different elements with regulators. So there's general guidance around the amount of efficacy data that really has started off many statistical discussions in planning trials. So in particular, there's some guidance around combination products. This is more recent guidance, and both the FDA and EMA have guidance around this. And so co-development of 2 or more new investigational drugs for use in combination. And there's quite specific guidance around the types of trials and the number of trials and the kinds of aspects that you need to deal with in a development program. We'll talk a little bit more about combination products when we get onto multiplicity a little bit later. So let's move on now to meta-analysis and single pivotal trials. You read in the guidance earlier that there's a requirement, but usually for more than one adequately well-controlled trial, but occasionally there's a possibility to do a single pivotal trial. And so meta-analysis and single pivotal trials, there's some guidance in this area. The EMA guidance talks about applications with meta-analysis or a single pivotal trial. And there's also guidance in the FDA about integrated summaries of effectiveness and safety. And then the meta-analyses of randomized controlled trials to evaluate the safety of products as well. So if you're in this area, you do any form of meta-analysis, this actually is quite an interesting read. So if you're not in the pharmaceutical area at all or into drugs, it's actually quite interesting if you're involved in meta-analysis, just to read through some of these documents to understand the regulatory review in drug development. So there are some prerequisites for accepting a meta-analysis for an approval. And the guidance between them go through major areas like in the meta-analysis, some studies are clearly positive. Inconclusive studies should show positive trends, so there should be no individual trial in the wrong direction. No major heterogeneity. A pooled confidence interval of treatment effect is well away from 0 or unity. And that biased trial selection is unlikely. So sensitivity analyses demonstrating robustness and looking at bias are fairly commonplace. And if you're in the world of meta-analysis, you'll be used to assessing this in your analyses anyway. So moving on about when not to have a single pivotal trial. Sorry about the build here. If you are having a single pivotal study and you're in one of these areas, then you have to be really careful because you're going to attract quite a lot of review criticism from regulators. So if you're in an area where there's an unknown mechanism of action or a new pharmacological principle, then having a single pivotal study is probably off the cards. Also, if your phase 1 and 2 data are unconvincing, you have a therapeutic area with a history of failed trials, and so on and so on. So I think that this does give guidance about when not to have a single pivotal study. Otherwise, you can look to do one. I'm sorry about the build here. That was unexpected. Okay. So prerequisites, if you are going to do a single pivotal trial, that you need to have internal and external validity. The observed treatment effect must be really clinically valuable. 5% level for type 1 error is not usually sufficient. And so typically we would look for an alpha level of 1% or less to demonstrate a clear efficacy in a single pivotal trial. I mean, you've only got one study to demonstrate your effect, so it has to be extremely clear. And it also has to be internally consistent among subgroups. And if you're in a composite endpoint, different components of the composite need to be in the right direction, and so on. No centers should dominate the result, and it should be completely plausible hypothesis that you're looking at. So in terms of validity, again, I apologize for the build. It actually goes through what it means by internal validity and external validity. And actually, this is quite a nice discussion because it talks about what guidance means in terms of internal validity for a trial and what guidance means in terms of external validity for a trial. So it's quite nice to see that this is laid out, and you can look through these points at your leisure a bit later. So in terms of the summary for meta-analysis and single pivotal trial, applications based on meta-analysis and single pivotal study are actually quite similar. And these are the aspects that need to be dealt with. They need to be well planned, convincing, confirmatory, no dominating subgroups, and very robust results. So this sort of summarizes the general guidance. If this is an area that you're working in, I really recommend looking at those guidance documents, having a read about what the pharmaceutical industry think about those areas. I'm now going to move on to the next topic for additional guidance, and that is non-inferiority. And in fact, there's a whole host of guidelines that are to do with non-inferiority. And then more recently with biosimilar products, there are definitions of biosimilarity, and in particular interchangeability. So I want to talk about these together. But really it started off with the concept of non-inferiority studies and about how regulators wish to interact with sponsors when designing a non-inferiority study. So there's a few key documents here. The EMA have one on how you can start a non-inferiority study and then switch to a superiority study without any penalty. But also gives really good guidelines on the choice of the non-inferiority margin, which actually is the big discussion when we're designing a non-inferiority study. The FDA have their guideline too, around non-inferiority trials to establish effectiveness. And these two guidances don't actually line up well. And in fact, the guideline on the choice of non-inferiority margin now reflects two approaches to setting non-inferiority margins. So again, if you're in a non-inferiority design setting, setting the non-inferiority margin, is good to look at those two documents to try and walk that line between setting a good non-inferiority margin. There's also further guidance about demonstrating biosimilarity to a reference product, and this comes from the development of biosimilar products and biobetter products. And also, if the biosimilar is to be used interchangeably with a reference product, the FDA have a guidance around demonstrating interchangeability, and actually have some quite specific guidances around how to do that. So these are areas that many of us know well, but I will go and just summarize these at a high level. Superiority studies are studies that should be based on statistically significant differences between treatments and demonstrating that, but the clinical relevance is actually a matter of judgment. And there's guidance around planning these. Many of us know these guidances really quite well. Moving on to equivalence, and in particular bioequivalence and biosimilarity. Here it's required that the equivalence margins are predefined and set ahead of time. And then the treatment difference, so the objective is to estimate that treatment difference, and the whole of the confidence interval for the treatment difference should lay between the equivalence margins. And so there's guidance that exists about how to select equivalence margins in the bioequivalence setting. This is quite regimented and quite specific about how to demonstrate bioequivalence of pharmacokinetics. But actually, in terms of clinical equivalence, the guidance is actually quite instructive too. Moving on to non-inferiority, this is a design that I see quite a lot. The demonstration is that a new treatment is no worse than another, but presumably has another benefit, usually safety or frequency or some measure of dosing is much better than what's already there. As before, the non-inferiority margin should be predefined, and as I mentioned, a lot of guidance around how to set that non-inferiority margin. The EMA guidance is quite interesting when you read this, because the guidance says that ideally a non-inferiority study should be designed with 3 arms. So your new product, a reference product, and the placebo. Of course, if there are acceptable treatments that are out there as reference product, you can't really deny a patient treatment, so you actually can't give the placebo. But I think that's an idealistic design, and they do say that they accept 2 arms without a placebo, which are the vast majority of the non-inferiority studies that we see. Again, the EMA guidance and the FDA guidance are good reads in this space. And both regulatory bodies that I refer to here accept the possibility to switch to superiority if you've demonstrated non-inferiority, and the results show a very convincing difference. Let's move on to biosimilarity. Biosimilarity is a concept that's come about recently with biosimilar products and is demonstrated using a number of different aspects. One is structural assays of the drug, the functional assays, animal studies, and sometimes the clinical studies too. More recently, there have been biosimilar approvals without clinical studies, but they have demonstrated pharmacokinetic equivalence. Occasionally, clinical studies are required, and they should demonstrate that there are no clinically meaningful differences between the product and the reference. And again, there's guidance around there about how you go about doing that. Clinical studies should include PK and PD studies and immunogenicity assessments. Comparative clinical studies may be necessary with an equivalence objective and predefined equivalence margins. Extrapolation across indications is possible, and with a biosimilar product, it's the totality of the evidence that it's assessed, no individual study. And then there's the concept of interchangeability. So this is developing a new drug to be used interchangeably with another reference product. And interchangeability is actually another requirement in addition to biosimilarity. And this needs to be demonstrated in dedicated switching studies. And the guidance actually gives some examples of what an interchangeability study might look like. And you can see here that patients on a reference product are randomized between an arm that doesn't switch and an arm that does switch. And on the switching arm, patients are allowed to switch between the interchangeable product and the reference product. And some rules and guidances around the design of that are actually good to get to look at. Okay, so that's interchangeability. I'm going to change tack now and move on to subgroups and baseline covariates, which actually is a topic that's very relevant to almost anyone working in clinical trials. And there's actually some really good recent guidance here from EMA and FDA on the investigation of subgroups in confirmatory clinical trials. The FDA have one which is adjusting for covariates in randomized trials for drugs with continuous outcomes. But there's actually a lot of good concepts in here. And again, I'm just going to go through some of those that I pulled out of these two guidances. So one is around subgroups. And the guidance talks about 3 scenarios that you might find yourself in. So the first scenario is the one that we hope we're always in, which is that you've declared overall significance across all the subgroups and the therapeutic efficacy has absolutely been demonstrated. And in that case, the clinical data are overall statistically persuasive with the therapeutic efficacy demonstrated in the population. And the purpose of subgroup analyses then is to verify the conclusions applied consistently across all subgroups. So you already have the positive effect that's therapeutically demonstrated, and you're now looking at consistency of effect. The second situation is where there's overall significance but borderline efficacy. And there, the clinical data presented are statistically persuasive in the primary analysis, but the therapeutic efficacy or risk benefit is borderline. And what's interesting here is to identify post hoc a subgroup where efficacy and risk benefit is convincing, and therefore some that might be unconvincing. And so that's a different situation that regulatory reviewers find themselves in. The last one then is when there's no overall significant result. So the overall data fail to establish statistically persuasive evidence, but there's interest in identifying a subgroup where a relevant treatment effect and compelling evidence of a favorable risk benefit may be assessed. And typically in this space, this is generally considered hypothesis generating rather than confirmatory. So having an overall non-significant effect but demonstrating a significant effect in a subgroup is usually considered as hypothesis generating but then confirmatory. So the overall concepts in subgroups then, and there are lots of topics that are talked about in these guidelines. One of them is about the credibility of the subgroup. So any subgroup that is predefined where there is biological plausibility and there is some scientific rationale for that subgroup is going to be more credible. A subgroup that comes about after dredging the analysis, looking at 300 or so different subgroups and finding the one that's significant, is going to be much less credible. So it has to be consistent among the endpoints, and if there's replication in a couple of different studies of the same subgroup, then it's going to be much more plausible. So these are concepts that are talked about in the guidance around subgroup analyses. So assessing subgroups then, in the first situation, if there is biological plausibility, or if the subgroup effect has been replicated in other studies, then the subgroup effect is likely to be more credible. In the situation where there's borderline efficacy, the subgroup is planned, pharmacological rationale exists, and the treatment effect is larger than the overall result and is replicated in the other trials, then it is much more credible. If the situation that there's no overall significance, then more trials will usually be conducted to look into that space. Although there's always a get out here, and they do say only in rare instances there may be a basis for pursuing regulatory approval without conducting additional studies. I think that is extremely rare and may only occur in rare diseases where doing additional studies just may not be possible. So the guidance talks about implications for study planning. For subgroups in study planning, it's always good to review whether you need to have separate trials in each subgroup. That may actually be necessary. If not, then the need to stratify the randomization by those subgroups is recommended. It's also good to assess whether you need to sample size separately within the strata to produce significant results in each strata, and occasionally that needs to be done. There are some disease areas where that is necessary. And you should always plan to examine exploratory subgroups in the analysis. And it begins to talk about the concept of predefined subgroups and exploratory subgroups in your planning. Around baseline covariates, there's some interestingly specific guidance around the analysis of baseline covariates in the guidance. I wanted to bring some of these out. So there are some comments around using analysis of covariance to adjust for differences between treatment groups in relevant baseline variables to improve the power of significance test and the precision of estimates. So it's appreciated that this actually is of an advantage. However, it does say you should not use ANCOVA to adjust for variables that might be affected by the treatment. So anything that's post-baseline or might be affected by the treatment, and there's a strong recommendation not to adjust for that covariate in the analysis. These baseline covariates should be prospectively defined in the protocol and the analysis plan. But it also says that the presence of an interaction does not invalidate analysis of covariance as a method for estimating and testing an overall treatment effect. And this is where it begins to talk about the difference between a qualitative interaction and a quantitative interaction. Quantitative interactions, you may still get positive outcomes of the treatments at different levels of the baseline covariate. It's just the size of the treatment effect may differ, and that's not usually a problem. So there's some quite specific guidances around the analysis using baseline covariates, which again, we nearly always get into when we're planning and analysing results from trials. And again, it's just good to understand what the pharmaceutical regulators are specifying in this area. So that's enough about subgroups. Let's talk about data monitoring committees. So we talked a little about data monitoring committees in our last session, where it came up in ICH E9. But clearly there's not enough detail in E9. So both regulators have given more guidance on data monitoring committees and how to operate them. So general guidance on data monitoring committees from EMA, and then more of a procedural guidance from FDA on establishing and operating a DMC. So the overall guidance talks about not all trials needing DMCs, and it gives examples of trials that do not necessarily need it, and then talks about areas where DMCs are usually needed, and talks about life-threatening diseases, pediatric or mentally disabled populations, where there's a strong suspicion that the treatment may provide harm to patients, and studies with pre-planned interim analyses for early stopping or modifications of study design, adaptive design. So these are usual in this space. Most trials now have planned interim analyses for adaptation of some sort or another, even if it's for early stopping or futility and so on. So we find that most confirmatory trials with an interim analysis do actually need a data monitoring committee. So the responsibilities of the data monitoring committee are laid out in these documents, which data is reviewed, how they make recommendations to the sponsor, and about ensuring integrity and credibility of the trial. This last point is actually taken extremely seriously. The big worry about interim analyses is the inflation of type 1 error, of course, but also operational bias and bringing in bias into the conduct of the study through the knowledge of the results. And so processes around data monitoring committees should be documented, written down and signed, using policies to avoid dissemination of interim data prior to unblinding. So this is a strong lead from reviewers on this. It talks about timing of the DMC committee meetings, the composition, and it does say that there should be a statistician with expertise on the data monitoring committee, and that the number of members should be limited. Talks about qualification of DMC members and independence of the data monitoring committee. In particular, no financial interest in the outcome of the trial, and no non-financial conflict of interest. And these are very important in the establishing a data monitoring committee and finding members to work there. So the guidance also talks about working procedures, and there's a lot of detail in the FDA guidance about working procedures. And it talks here about the existence of a data monitoring committee charter that's in place before the trial starts, and it should include all the following items. So if you're in the business of writing a DMC charter anytime soon, you might want to take a look through this guidance to see how it affects what you're about to do. And then talks about the implications for study analysis. The major issues are inflation of type 1 error, possible bias in the future conduct of the trial. And then also talks about the type of monitoring and what you need to be concerned with. So the primary outcome, type 1 error control should be detailed in the protocol. They may still assess the primary outcome to assess risk benefit, and the impact of the type 1 error should be properly taken into account in safety as well. The role of a data monitoring committee is actually relatively complex, and really they're there to assess not only efficacy and safety together, but also the risk benefit of the situation, which is why they need to have that experience there. Design modifications have to be described in the protocol, and the DMC members need to understand clearly the statistical plans for advising on those modifications. And futility is very common as an action for data monitoring committees as well. And again, it talks about dissemination of trial results and having a really good communication plan in the charter for how the DMC communicate the recommendations to sponsors is critical. Okay, so that's sort of a summary of the guidance on data monitoring committees. Now I'm going to move on to a big area, which is type 1 error and multiplicity. And there's a lot of guidance around multiplicity in the regulatory documents, as you can imagine. And some of them are actually quite good reads as well. So the EMA have two. They have an old one, which is points to consider on multiplicity issues in clinical trials that came out some time ago. And then a more recent one on guideline on multiplicity issues in clinical trials is actually quite a good read. as is the FDA one, which is concerned around multiple endpoints in clinical trials and how to adjust for multiplicity in those spaces. So the guidance together talks about when to adjust. It talks about multiplicity being present in virtually all trials that we do, and does specify when adjustment is not needed. And then says, if you're not in those situations, adjustment for study-wise type 1 error is required. And if not handled, the rate of false positive conclusions may be inflated to unacceptable levels. Most interactions with regulatory bodies that I'm involved with, there's usually a big discussion around the false positive rate in the trial and the type 1 error and how it's controlled and the methodology that's being used and whether we've taken account of all of the multiplicity aspects of the trial. There are a number of methods that exist and do depend on the type of multiplicity. And the FDA guidance recently gives a really good summary of some of the methodologies that exist for multiplicity of different endpoints, and is actually quite instructional. If you've not come across this area before, it's a good document to read first, actually. So it talks about types of multiplicity, so multiple primary variables, multiple analysis sets, multiple methods of analysis, multiplicity and safety variables, and actually when there's more than 2 treatment arms. So there are a vast number of different areas of multiplicity that the guidance says that we should take care of. And in particular, I'm going to pull out 3 areas. One of them is the efficacy for a fixed combination. And this is relatively common, where there is a combination product, AB, and you may have the monotherapy A and the monotherapy B, and you may even have a placebo, although that is unusual if there's already monotherapies around. And it says here that multiplicity adjustment is not necessary. So if you've demonstrated superiority of the combination to the monotherapy A and the superiority of the combination to the monotherapy B, then you don't need to adjust those for multiple comparisons. So in a multiple dose factorial design for a combination, the global test strategy can be used to demonstrate superiority. But if you have a dose recommendation to be done, then that area does need the multiplicity adjustment. So this is an interesting area for fixed combinations, which actually is an area for not adjusting for multiplicity. In dose response studies, usually in phase 2 studies, formal adjustment for multiplicity here is not normally required, where estimation is the aim. But actually, a positive correlation of improving clinical outcome with a dose is valuable. So if you can demonstrate the treatment effect, then that's actually valuable for you. If you're in a situation where you have a confirmatory trial with more than 1 dose, then you do need to adjust for multiplicity. In fact, it's mandatory. So this is another area where quite specific guidance for multiplicity. The area of composite variables is quite common in most diseases that we work with, where the composite endpoint is the primary endpoint. There's actually guidance around composite variables, and that the treatment effect should affect all components beneficially, i.e. not negatively. The clinically more important components should not be affected negatively, and any effect on a component that is to appear on the label should be supported by the data. So there's quite specific guidance around composite variables and what a robust result needs to look like. So I'm just going to talk about this FDA document that talks about multiple endpoints. If you're in the area of multiple endpoints and understanding or wanting to understand multiplicity in this area, this actually recent document from FDA is really very good because it takes you through a lot of the common methodologies for dealing with multiplicity and multiple endpoints. And it does give specific details and some examples actually of all of these approaches. And what it does is it gives the FDA's view and understanding of what these methodologies are and gives examples of how to use them. So again, this is a really, really good read if you're into learning more about multiplicity. Okay, so we've come to the end of the overview of the additional guidance. And I'm going to turn now to the second part of today's talk, which is around adaptive design guidance. And we mentioned last time that there's a new guidance, E20, that's being worked on right now. And E20 is guidance around adaptive designs. There are currently a number of guidances that are relatively recent around adaptive design that have come from the regulatory groups. And these are those. So adaptive design for clinical trial, a reflection paper from the EMA, enrichment strategies for clinical trials, and more recently, master protocols and guidance around master protocols and efficient clinical trial design strategies to expedite development, in particular of oncology products. More recently, the Complex Innovative Design initiative from the FDA has come about, and they formed a project of sharing expertise with FDA and sponsors when there is a complex innovative design, and I'll talk about what that is shortly. But there's also guidance about how to interact with FDA on the complex innovative trial design. So if you're in the area of a complex innovative trial design and you want to use that in a confirmatory setting for your particular drug that you're working on, then there's a strong recommendation to interact with the FDA on this, and they will help you in the design and the consideration of the design that you're going to use for your confirmatory trial. Nearly all of them deal with the following principles. Controlling the chance of an erroneous outcome. Sorry about the typo there. And this is all around decision errors. This is false positives, false negatives, type 1 error, and so on. And it doesn't talk about type 1 error specifically because it does allow for the fact that Bayesian approaches can be used. Estimation of treatment effects. In most adaptive designs, particularly where there's selection involved, the estimation could be biased, and there are methodologies now being developed to provide adequate estimation of treatment effects in different areas of adaptive design. It talks about trial planning and pre-specified adaptations. And also the big one is around maintaining trial conduct and integrity. So minimizing bias, particularly operational biases. And this is big in the guidance around adaptive designs. So in terms of adaptive designs around comparative data, there are now some adaptive designs that are said to have well-known characteristics. So there are some confirmatory trial adaptive designs that the regulatory bodies are used to seeing now and do accept if they recognize the particular kind of adaptation as being one that is well-known. And there are a number of those. So group sequential designs are clearly one of those. Sample size re-estimation is another one, unblinded sample size re-estimation. Patient population, so this is enrichment trials. Treatment arm selection, so this could be dose selection, for instance, and changing patient allocations as we go through. In particular, adaptive randomization is not yet a well-known characteristic trial, but it's nevertheless accepted as an adaptation for comparative data. And endpoint selection is another one which is less accepted, so changing the endpoint and adapting the choice of the endpoint. So you have to be very clear about when that's being done in a trial, and of course it all has to be predefined. But that is one of the not so well-known adaptations. And then of course there are designs that feature a number of combinations of those. So patient enrichment followed by sample size re-estimation is something that I see and in our work here at Cytel. Work here at Cytel, we advise and guide on design and simulation of many of these adaptations. And so we are quite used to understanding regulatory stances in most of these areas. The adaptive design special considerations. So as you look through the documentation, there are special considerations in each of these areas. There's a lot of discussion around simulations and the role of simulations in understanding operation characteristics of trials. Bayesian adaptive designs, I'll talk about that specifically. Adaptations in time to event settings, Secondary endpoints, adaptive design in early phase is quite common. Unplanned design changes based on comparative interim results is an area of special consideration. And design changes based on information from a source external from the trial as well. So if you're in any of these situations, the guidance documents calls this a special consideration, and it's good to understand the different areas and different guidances that you may get. So Bayesian adaptive design is referred to in these documents, and the use of predictive statistical modeling to govern the timing and decision rules for interim analyses. So common ones are the use of assumed dose-response relationships, where there are prior information around the parameters of the dose-response relationship to govern dose escalation and selection. In oncology dose escalation, this is common, and more so with bringing in dose-response models, and Bayesian versions of those are beginning to be accepted for dose selection in phase 2. Explicit borrowing of information from external or historical sources to improve the efficiency of the trial is a real current topic. Synthetic control arms and the use of historical data to formally bring into the comparison of the active against the concurrent control is an area that we see quite a lot of work in at the moment, particularly in the rare disease space. And this is becoming more and more accepted. I think we're a little way off this, having been considered an adaptation with well-known characteristics, but there's a lot of work going on in this area right now. And in particular, the use of posterior probability distributions to form trial success criteria is becoming more accepted in this space. So there's good guidance around enrichment designs, and actually good examples given in cardiovascular oncology, pulmonary and neurology studies, and talks about potential strategies for different areas, and it accepts that there are different categories for enrichment. One is to decrease variability, prognostic and predictive enrichment are others, and actually goes to actually provide examples of designs that you may wish to use under different circumstances. So if there's no possible effect in the marker negatives, you might go for the top design. If there's a possible effect in the marker negatives, you might go for the middle design. And the marker cannot be assessed before randomization of the marker negative patients. You might go for the bottom design. And it talks about situations where you go with different designs. There's actually a lot of discussion around how many marker negative patients that you need to have in your trials before you submit for an application. And there's some guidance here around how many marker negative patients that you would need in your trial or in your application. And it depends on these different needs. I'm going to turn now to master protocols. And the guidance talks about the different types of master protocols, basket, umbrella, and platform studies, and gives definitions of these. But actually gives other aspects. So the use of common control arms, novel combinations within your platform trial, studies with drugs targeting multiple biomarkers, adding and stopping treatment arms, the role of the independent data monitoring committee, which actually is quite a big job in these trials, developing biomarkers during the trial, and so on. And the contents of the IND submission and interactions with FDA if you're involved in a master protocol. It also talks about statistical considerations in master protocols. So if you have a single-arm, non-randomized estimation aspect in your basket study, for instance, then use sample size to rule out futility, and it talks about the requirements in the analysis plan for those. If you're in an umbrella design, so this is a single disease with many different treatment options, it strongly recommends the use of a common control arm. And master protocols employing adaptive Bayesian designs, that the analysis plan turns into quite a big document and should contain all the details, including all the analyses and implementation details of Bayesian and other methods. So there are a lot of statistical details for master protocols that need to be worked out. These recent guidances do actually give some good examples, and they give examples of a basket study, an umbrella study, and a complex master protocol. and talks about different aspects of the statistical considerations using these examples. So it is actually quite good to understand this if you're in the world of master protocols. Complex innovative design is one of the last things that I'll talk about. And so the Complex Innovative Design Initiative that's been set up by the FDA actually talks a lot about complex innovative design. And it defines a complex innovative design as a design that you can only understand the operating characteristics for under trial simulation. And that's really a definition. So it turns into an adaptive design that is not well known in terms of characteristics. And includes trial designs that have been rarely or never been used before, requiring simulations to understand trial operating characteristics. Bayesian proposals do need to include robust discussion of prior distributions and decision criteria. And these are characterized by having many required simulations to understand the impact of these. And if you're designing one of these, actually you should be very interested in this anyway before you go near a regulatory body with any of these. And it does actually give examples. So one is incorporating phase 2 data into phase 3 trials. And then sequential multiple assignment randomized trials. So these are the examples that are given. So complex innovative design is becoming more and more frequent in terms of particularly working in rare diseases and in oncology. And we see a number of different complex innovative designs that come our way as we give advice and guidance to sponsors. So we're coming near to the end of the time, and I just wanted to review back on the content of the course that we've done over these last 2 sessions. In the first session, we concentrated on ICH framework, good clinical practice, the relevant ICH e-documents particularly for statistics, but there are a large number of other e-documents which we spoke about before that you may be interested in reading through. And as I mentioned before, it's very hard to give any detail on any one of these guidance documents, but I hope what I've done is given you an overview and so that you know where to look in the future as you go through these. What we've just done is look through the additional guidances and the guidances around adaptive trial guidance. And again, as you look back over these slides or you look at this video again, if you have any questions, please do bring them to us and we will try to answer those and post answers on the ACTA website. So Anisha, I think I'm done. And we do have a short amount of time if there are any questions.
Thank you very much, James. I do have one question from Himal. So the question is, can I have a link to the EMA where James said they have recently published guidance for using real-world evidence on clinical trials? The one where James said they are asking for feedback.
Yep, so that's available on the EMA website. So if you click the link that I provided on one of the earlier slides, you should be able to find that. And if you search for real-world evidence, it will come up. If you have any difficulties finding it, please let me know, and I can send you the specific link.
Thank you, James. The second question is from Julie Marsh. The question is, do investigator-led trials need to collect safety data if the products are already approved?
So this is all around assessing the risk-benefit. So if there's a really good understanding of the risk, then collecting safety data, I feel, is still useful because what you want to do is assess the risk in the specific trial situation that you're in. And you may see unusual or conflicting or uncertain efficacy results. And what you're left doing in that situation is saying, okay, so here's the efficacy, what is the risk? And it's usually always better to understand the risk in terms of data coming from that trial to assess that risk benefit in that trial situation. I think the amount of safety data and that you collect, that's an area for discussion. So you may collect only serious events and so on, but I think collecting some safety data from those trials is very useful to assess the risk benefit in the trial that you've done. I think if you were to conduct a trial and collected no safety data, that would be almost impossible to assess the risk benefit in that population.
Thanks, James. The next question is from Kylie. Are there any areas where you think the official guidance is wrong or outdated?
I think if the official guidance usually comes from committees, and in most of the guidance documents, they do ask for review. So there's a review stage. So we find that the first versions of draft guidances that come out, there's usually big areas of contention in them. But by the time they've gone through the review process, they've been watered down or taken out or discussed in a different way. So I find that the guidance documents are usually quite considered and considered by wide review. So we don't get guidances that are dictatorial that come out without the process of having gone through this discussion stage. So I think the short answer is no. Some of them are that you might not agree with, but they've all been considered by a large group of specialists. I don't think there's any that I completely disagree with, actually. I might have differences of opinion in some technical areas, but I think in most areas that they are very good and very well written, and good guidances, and good to understand before you get into designing trials.
Thanks, James. I think that brings us to the end of the Q&A. We do have an evaluation for the webinar, which should show up after the webinar ends. Please take a moment to provide your valuable feedback. Thank you again, James, for your valuable time. On behalf of the Australian Clinical Trials Alliance and our presenter, James, thank you for joining us today, and enjoy the rest of your day. Thank you all.